this video, I'll talk through the 2024 Higher Human Biology past paper and explain how I would approach every question. Starting off in the multiple choice, we're asked about making red blood cells from bone marrow. One way of approaching this is to look at the two combinations of words, where we have multipotent is the word used to describe tissue stem cells, and pluripotent is used to describe embryonic stem cells. One way of narrowing it down to A and D is they are correct statements. We're asked about making red blood cells from bone marrow. As these are not found in an embryo, this is going to be an adult tissue stem cell, which is multipotent. Next one is asking us to deal with the number of bases in a DNA molecule. One thing to remember is that if we have 1, 2, 4, 5 of adenine, we'll also have 1, 2, 4, 5 of thymine. Then telling us that there is a 3:2 ratio of thymine to guanine, and we need to work out how many guanines there would be. Now, a couple of options for doing this. My preferred way, if we want to go from 3 to 2, one thing we can do is if we first divide by 3, that will bring us down to what 1 would be. Then we times by 2, take us up to what 2 is. If we do those same operations, 2, 1, 2, 4, 5. That gives us 830 as our answer of C. Another option, what you could do, well, 3 to 2 is the same as 1.5 to 1. So divide both of them by 2. And then if we have 12, 4, 5, we want to see what the other number is. Well, if we do that, divided by 1.5, 12, 4, 5, divided by 1.5. Also gives you 830, because again we're dividing by 3 and times it by 2. Sure, there are other ways of dealing with that as well, just whatever way works best for you and those kind of values. Next question is dealing with a catabolic reaction. If you've ever met a cat, you'll know that they are much more likely to break down materials rather than build them up. That means we're not going from small to large, we're going from large to small. And the reason to do this is they are releasing excess energy, and we then release energy. Or the answer of D. If it was an anabolic one, we would be going from small to large. That would mean you'd need energy, so anabolic would be A, would be the correct answer there. We are investigating the rate of respiration of an organism. Nice little snail. We want to do a suitable control. Now the control is trying to prove, is the snail required for this reaction to take place? So do we need the snail for respiration? We want to make sure we're keeping all the rest of the setup the same. We want to replace the snail with a non-living version. If you do more organisms, you're repeating the experiment, that can make your results more reliable. If you use a wider range of temperatures, you get more data and you can see what happens along a further range of the temperatures. So it might have an unexpected effect at higher temperatures or lower temperatures. The glass bead, we are removing the organism, replacing it with non-living version. C would be a correct answer there. Answer D would let us see what happens without the solution, and then we could see if that was needed for the reaction to take place. In this one though, we're asked about the organism. We want to do it with and without the organism. For question 5, we're asked about personalised medicines and picking the most effective drugs for that patient. Pharmacies are places people go to get drugs prescribed to them. Pharmacogenetics would deal with that one. Bioinformatics is when you use computers to study and analyse the data from genetic information. Human genomics would be the study of the entire genetic material in a human, so the Human Genome Project. Genomic sequencing, you look at the order of the bases in the DNA and use them to compare things like paternity. You look at potential for developing diseases and evolutionary connections between different organisms for that. Drugs, pharmacy, pharmacogenetics gets us C there. In this one, we're asked about different types of inhibition. Okay, so first thing is inhibition due to substance S. Well, substance S is an end product which is going back to stop an enzyme earlier in the pathway. That is feedback inhibition. Next one, we've got competitive and non-competitive inhibitors. When using a competitive inhibitor, if you have more of the substrate, there is a greater chance of one of the substrate molecules binding to the active site before the inhibitor can block it. meaning that it is reversed at higher concentrations of substrate. And non-competitive, because the shape of the active site has been altered, the substrate cannot bind to it because it's no longer complementary. Doesn't matter how many of the substrate molecules you have, they won't be able to bind because the shapes aren't matching. If the effects are reduced by the substrate concentration, that means that we're dealing with competitive inhibition, and that gives A as our correct answer for that one. Next one, if you have got a premature stop codon, you've got a shorter protein, that protein will be a nonsense mutation. When we're dealing with missense, we'll have a different amino acid in the chain. It could have no effect, it might have a subtle effect on it. It wouldn't cause a shorter protein to be produced. When we're dealing with splice site, it affects exons and introns. Which isn't what we want here. Frame shift, all amino acids after the mutation are affected. And that's not what they're talking about in this question. Number 8, we're asked about the different types of mutations that have taken place. Let's follow the stripy chromosomes. First of all, we have lost a piece of the chromosome which has ended up on a different chromosome here. So we've moved information from one pair of chromosomes to a different pair of chromosomes. That is translocation. That leads me to be A or D. And then the other one that's happened is the grey one here has lost genetic information. We have had a deletion for that there. If it was inversion, it would have swapped around the order of the genes. In this one, we're talking about fermentation. And here they're saying once the oxygen has been replenished, then we're reversing that conversion. During fermentation, pyruvate is changed into lactate. So here we're going to be going backwards to making pyruvate. And pyruvate was formed when glucose was broken down. That would make our best answer A for that one. Okay, in this one, we're talking about PCR, where every cycle of PCR doubles the number of DNA molecules. And we've got a section of DNA. Okay, starting off with 1, we then double it to 2, and onwards to 4, 8, 16, 32, 64, 128, and then 256. We are looking at the number of cycles, which just means how many jumps have we done. So every arrow is a cycle. We have 1, 2, 3, 4, 5, 6, 7, 8 cycles. And the question, though, isn't asking us for how many cycles there are, how many temperature changes are there. And it says that there are 3 temperature changes per cycle, because we heat it up to break the hydrogen bonds between the strands, cool to allow primers to bind to target sequences, and then warm it up again to allow the DNA polymerase to replicate the DNA. So if one cycle has 3 changes, well then 8 cycles will have 3 times 8, which gives you 24 changes. So a wee bit of a change from what they might typically ask about how many cycles. Every cycle has 3 temperature changes, and that's our answer there. In this one, we're dealing with making of sperm. So a couple of things to deal with this here. So sperm is produced in the seminiferous tubules. And the other thing that's produced is testosterone in the interstitial cells. And that brings us down, only with these ones here. Sperm is one of the gametes, which is haploid, has half the information for the new individual. And they are produced by meiosis, makes the gametes for that. Mitosis produces identical daughter cells. And here we're going from the germline cells to the gametes. So that's not making identical copies. That is in fact meiosis that would have for answer D in number 11. Next one, with the different types of contraceptive pills, they all have different mechanisms by which they prevent pregnancy. We are here dealing with the progesterone only pill that promotes the thick mucus blocking the sperm. Sperm is going to enter through the vagina. We are going to be dealing with, that means the answer would be structure D. In this question, we're dealing with inheritance of a recessive condition. They're asking us to identify who proves it's not sex-linked condition. What I would do is I would go through it as if it was sex-linked and see where the pattern breaks down. Now, for females, there are 3 combinations of alleles we could have. We could have X big T, X big T, X big T, X little t, X little t, X little t. That one. For males, there are only 2 options. We can have X big T, Y, and X little t, Y. So it's only carried on the X chromosome, and that's a different combination you can have. If the person was affected, they must have only the little versions, only little t. That means if they've got 2 little t's as they're female, they will be affected. And if they don't, if they're male, they'd have X little t, Y. For that. For an unaffected male, they must be X big T, Y. And then an unaffected female could either have 2 big letters or 1 of each. So they might be homozygous dominant or heterozygous. Now they are telling us that we've got a grandchild of a male with the condition. So the affected male has to be X little t, Y. And then it's saying that the female is homozygous. So she has to be in this situation X big T, X big T. If we look at our Punnett square, we have the different crosses here. We would have X big T, X little t, X big T, X little t, X big T, Y, X big T, Y. So we would have all unaffected because they've all got at least 1 big T. And that matches up the same there. So that would be okay. So that this line here, P and Q, could work from this inheritance pattern. If we then go on and look at the next set, which would be this pairing here, well, T has to be X big T, X little t, because that's a female coming from the combination of the parents. So they have to have 1 of each. And then an unaffected male has to be big T and Y. So in this second Punnett square, we've now got X big T, X little t, X big T, and Y. Do all the combinations. X T, big T, X big T, X T, and Y, X little t, Y. So all the females would be unaffected, but we have got an affected female there. This means that this person R shows that this pattern wouldn't work. You could still get an unaffected female. That is a potential outcome, but you cannot get an affected female from that combination there. There are some ways of shortcutting these kind of questions where if we know that the father at the second stage has got a dominant allele, they have to pass that on to both their daughters, because if they passed on a Y, their daughter would be their son. So it has to pass on X big T, which means, worst case scenario, the daughter has 1 of each, which would still mean they don't have the condition. So that's not going to prove that as well. So an unaffected father having a baby with an unaffected mother will not produce an affected female for that one. And that works out as answer C. Next one, talking about artificial insemination, where the fertilization happens outside of the body. There's me clearly not read the question properly. We're not talking about IVF, which is where my head went first of all. It's talking about artificial insemination, where sperm is inserted into the reproductive tract, giving it a better chance of fertilizing. So that means that you would get multiple samples to give them the best chance to get enough viable sperm to be able to perform the fertilization. The other ones, you're dealing with IVF, and that threw me off at first. Make sure you read the question properly and you'll hopefully avoid that. Here we've got an inherited disorder caused by mutation that affects a reaction in the process. Type of mutation in the reaction. Now, this is probably one that if I had went through the notes a bit more carefully, that will be an example that we are supposed to remember. Let's say that, like me, you don't have it off the top of your head. Now, my thought process for this one is if it's deletion, it's a frame shift mutation which affects multiple, it affects all the amino acids after the mutation. And that feels like it wouldn't just then affect one part of the pathway. With my answer there. Now, based off its name, I know it's to do with phenylalanine. I know it's found in it's in high protein foods. And it's tested for by the heel prick test to see if the baby has that. They can make sure they have a diet that avoids phenylalanine products for that. So it's not able to get rid of that. It's the build-up of chemicals that come from the phenylalanine. It's going to affect that one as well. So that'd be my kind of like muddling way through of getting it. Next up, we're dealing with cardiac output. Cardiac output is going to be the heart rate times the stroke volume. So how many times the heart beats and how much blood is moved at each time. We're asked about the increase after training. So before, that would be 60 times 0.07, and after, going to go 51 times 0.09, 4.2, 4.59, take them away from each other, we've increased by 0.39. And this one we're dealing with making a clot. First of all, clotting factors are released. That encourages the conversion of the inactive enzyme prothrombin turns into thrombin. After that, then fibrinogen is converted into fibrin, which then causes the build-up of the clot and stops the blood flow. For number 18, we're asked about BMI. That's going to be the body mass over the height squared. That's just something we just need to have memorized. Body squared, body mass. 18A for that one. Corpus callosum is the part of the brain that connects the two hemispheres. That sounds good to that. Localization isn't talking about a connection. It allows information to pass between the opposite sides but isn't processing it. And it's not to do with short term, long term, it's to do with the two hemispheres, two sides of the brain. That one is answered there. Cerebral cortex is to do with memory and conscious thoughts. So imagination would be part of that. Processing, yes. Balance would be the cerebellum. Because we're looking for the one that's not. C is the correct answer there. Next one, a recreational drug. So the reward pathway is to do with dopamine. And that would mean it's going to be A or B. An antagonist would block the dopamine receptors. That would stop it from working. So it's not going to be that one. If it blocks the reuptake, it means that the levels can increase and it's not going to go away. Okay, in this question, we're asked to predict the dopamine levels at 120 minutes. So dopamine is our dashed line. And we need to ensure that we are reading it off of the right hand axis for that. So what's happening to it? Well, it's going down. Now we're asked about 120 minutes. So on the right hand side, we must be jumping up in 5s, 5, 10, 15, 20, 25. That means that we are at 80 minutes, we're at 245. Then after 100 minutes, we're at 180. So in 20 minutes, we've gone down by 65%. And if we go in for another 20 minutes, we're going to again decrease by 65. So 180 take away 65 would give us 115%. And the reason that I've said it jumps by the same value is that it's going down steadily over the 20 minutes from 80 to 100. Another option, we could extend this line by the length of 20 minutes. That would be there. I run that up here. I keep it as straight as I can. And then extending on our line here. Try and get it through as best as possible. So roughly there. And then that comes off at just about 115. That part of the issue is if you see my wee purple, but I've not lined it up particularly well. But that, especially when you look at the jumps between the options, there's nothing really. The only one close to where my little blue line sitting is B, and that would give us our answer of that one as well. So extending the graph, extrapolating the data could work, or looking at how much it's jumping by as well would work. Always make sure you're taking it off the right hand side in this question. The other side is to do with serotonin, which we don't get asked about. In this one, we're talking about non-specific body defences. Mast cells produce histamine, which increases the blood flow to that area. And that's not specific phagocytes engulf and destroy pathogens. That's not a specific defense mechanism, that's a non-specific defense mechanism. Two different jobs, but one, the B lymphocytes release antibodies, and antibodies are specific to the pathogen that they're coming up against as well. 1 and 2 would be my answer. Next one, we're asked about independent variable, and this is what you, as the investigator, are changing. So what we're doing, investigation carried out into the use of different adjuvants. That means the type of adjuvant would be your independent variable. And adjuvant's the thing that enhances the immune response. Next one, we're asked about herd immunity, where we need 90% to have been successfully vaccinated. And one of them is going to be at most risk. So the further below 90 you are, the more at risk we're going to be. So we want to work out the percentages that we've got. So for A, work out your percentage, we want to look at, well, how many were vaccinated? That was 600,000. And then that is divided by the total population in that area. And then times it by 100 to turn it into a percentage. Next one. Now, I've again just found out a problem here. Well, I've misread that. So there were 600,000 vaccinated, but 10,000 were not successful. So that means that is going to actually be 10,000 less than that. So it'll be 5,900 times 100. Okay, the next one then for B would be 200,000. And then we've got 405,000 for C, and then 295,000. Then normal service is resumed. Divide them all by their populations and times by 100. What you should do here then is use your calculator, working out how many were successfully vaccinated, and then divide that by the total population times by 100 to turn them into percentages. And D has the lowest percentage vaccination, and that would mean that they would have a greater risk of outbreaks. Moving on to paper 2, we're asked about the DNA sequence in this test for detecting a virus. Complementary base sequence. It's still ATGC because we're still in DNA. T pairs with A pairs with T with A, C goes with G goes with C, and then A for the other one there. Next one, we're looking at temperatures to separate the strands. So this is where we heat it up to the highest temperature during PCR. Anywhere between 92 and 98. My preferred one is 95. And then 95 down to 55 up to 75. All fit within the values we're allowed. Primers bind to target sequences. Could describe it as being a starting position for the nucleotides to be joined onto, but binds to target sequences. Polymerase job to add the DNA nucleotides to build up the DNA for that one. Next up, other than diseases, other than diagnosing infections, what's other uses? You can use them to solve crimes, so forensic investigations, and you can also use it to see things like paternity tests. And we'll only ask for 2, but diagnosing genetic conditions. This one we're asked about organ transplants, transplant lists, state the number of lung transplants performed in 2020. The number of transplants performed is over on the right hand side. 2020 lungs. So white number performed. So we want to run that one across to there. So it's two little boxes up. Now, ways of doing this, so we can try out different numbers to see what happens. Another way you can do it is, well, there's 500 of a difference from there to there. That is for 10 boxes. Well, 500 is for 10 boxes, then 10 times smaller has to be for one box. So if that was true, then we go 50, and my answer would be 100. You can check again, 150, 200, 250, which fits in quite nicely for that. The other way you can do it is just go through them and go, okay, so try 1, doesn't work, 2, 10, wherever we get to it, it's 25, it's 50 at every box, meaning we've got 100 performed transplants. We have to calculate the percentage of individuals who received a kidney transplant after being on the transplant list at the start of 2020. Okay, now this is all 2020, so it's just this numbers here. So kidney transplant values. So we have got two things to look at. So the number of transplants performed would be, well, if it's 50 each time, then that will be 2200. 2200. So there was 2200 performed out of the waiting list. Now, thing to watch here is it's not the same scale over here. This time, every box has to be 100. And that gives us 5,600. Pop that in our calculator. Gives us 39.3%. For that, and you can allow you to round it a wee bit more or a wee bit less, and that would pick up the mark there. In this one, we're asked about how many transplants were performed in England during 2020. Okay, so the table says there was 47 transplants in every 1 million people. And we've got 57 million. So if it's 47 for every 1, well, we are 57 times bigger. So I want to times 47 by 57. Because if 1 million had this many, 57 has to have more for that. Popping that in. Gives 2679. I think of these ones as if you're in a shop or buying something where it's X amount of pounds per 100 grams, or you're buying one thing and if you want two, it's double. That's the kind of thought process I used to get that. Why are they always presented as per million? If there's different sizes of population, this allows a valid comparison between the countries. By virtue of having more people in the population, it's likely to have more, the numbers are likely to be higher. This one we're talking about enzyme activity, we're talking about catalase breaking down hydrogen peroxide into oxygen and water. This is likely for a lot of people to have been their assignment experiment, a very common one to be used because it works well and you can get good data from it. We're dealing with little beads, we're producing oxygen and catching it in the gas syringe. We're thinking about what things could have an effect on the results, so we need to control them to ensure that they don't have an effect. The one thing we're not allowed to talk about is the concentration of the inhibitor, the copper sulfate, because it's already mentioned that's what we're investigating. Enzymes are affected by things like temperature. That's my go-to one. Do they mention it up here? I don't see it. So temperature would be a good one there. pH, so how acidic or alkaline the solution is, as well, isn't mentioned. I'll put them together because that's quite common. things we can change are that. Now, when you're dealing with solutions, the volume or concentration of those solutions, and it doesn't say volumes of anything. So we could have the volume of inhibitor. That was your copper sulfate. Or volume concentration of hydrogen peroxide. Yeah. Other things could be the beads, how big the beads are, how many beads there are. It doesn't say that as well. So you've got quite a lot of options that aren't mentioned. If it's said up here, any of those ones, don't put them, put other ones that aren't mentioned. For that, why would we use immobilized beads rather than yeasts in solution? Well, the beads were getting placed into the inhibitor for a day and then moved across. You're trying to do that with a solution, it's going to be a lot more difficult to do. Makes it easier to move from inhibitor solution to peroxide. This is going to allow the inhibitor absorbed into the bead. Gives it time for it to do its inhibition. Next one, we're asked to do a line graph to show the average volume collected. So headings, you can just steal the descriptions from here. The thing that you are changing should go along the bottom. I'm going to deal with the average volume. That's in centimeters cubed. The highest concentration we've got to go up to is to 50. That feels to me like 5s would work. We can try them out. And that would, yeah, gets up to the maximum there. Average volume goes up to 82. And we've got how many boxes? 1, 2, 3, 4, 5, 6, 7, 8, 9 boxes. So 10s would work out there. Need to make sure that we're going to take up at least half the graph paper with our plots, which this definitely will. Like so. Now along the bottom, every box is going to be 1. Up and down the way, every box is going to be 2. So the first point we had was at 0, there was 82. Should be there. And then at 5, it was 68. Knock it down by 1. 10 was 45. Now 45 will be halfway between 50 and 40. So in there, so 42, 44, and then half a box would be 45. Works out there. Over to 25, we're at 25. So 25, as I said before, will be halfway in those two. And at 50, we're at 16. So 3 little boxes up from 10, 2 boxes down from 20. We then want our points plotted together. We just want to go from dot to dot, cross to cross, like so. Give a conclusion from this investigation. We were asked about the effect of the inhibitor concentration on the catalase activity. As the concentration has increased, the average volume of oxygen has gone down. We're not asked about that though, we're asked about the catalase activity. Well, the better the catalase is breaking down the hydrogen peroxide, the more oxygen is produced. So if we're making less oxygen, that means that the catalase activity is lowered. So as concentration of copper sulfate increases, catalase activity decreases. Next, we're asked about respiration. We're asked about glycolysis first of all. Same statement, so there's going to be more than one. So in glycolysis, glucose is broken down into pyruvate. That will be Y. Carbon dioxide released is happening in the citric acid cycle. Acetyl is part of the citric acid cycle. but we're needing the dehydrogenase enzymes to remove the hydrogen ions and electrons, pass them to NAD, moving that forward to the electron transport chain. Name the process that's broken down to form acetyl that binds to coenzyme A. Well, glucose goes to pyruvate, pyruvate into acetyl group. That must be pyruvate in there. Then there's oxaloacetate and acetyl joined to make citrate as our answer there. Okay, next one we're asked about why different mitochondria can produce different quantities of ATP. Why can the muscle ones get more than skin cells? Looking at the differences, their membranes have more folds. There's an increased surface area for the reactions to take place. And that is to do with the electron transport chain. In order to generate the ATP, hydrogen ions are pumped across the membrane. So if we've got a larger surface area of the membrane, that means more hydrogen can be pumped across the membrane. So endurance muscles will have more mitochondria for aerobic respiration. Aerobic respiration needs oxygen, so it's got a larger blood supply than the fast twitch. And then myoglobin helps store the oxygen. That would need that as well. Okay, next one, we're talking about the first half of the cycle, so the follicular phase. We're asked about FSH. Well, that stimulates the development of the follicle. And then the follicle releases estrogen. And estrogen stimulates thickening of the endometrium, ready for implantation. At day 14, the LH is going to cause the ovary to release an egg. Calculate how many times greater the concentration of progesterone at 25 compared to 15. So progesterone at 25, there, let's run that across. Okay, so what we're jumping up in here. Now, the way I'm looking at this, I'd probably do it in like every $2. This must be 5 for day 15. Progesterone all the way down here. So every $2 is 5, so 5. And a question we're asked about how many times bigger something is. How many times does the smaller thing fit in to the bigger thing? So 70 divided by 5 would give you 12 times. No, it wouldn't. It would give you 14 times bigger. 14 times 5, 70. That's the way to check. So how many times does the small one fit into the other one? The structures within the ovary. It produces progesterone, the corpus luteum. So as the follicle is developed into the corpus luteum, that's the then part that produces the progesterone. Evidence that they have not become pregnant. Their progesterone levels would stay high if they were pregnant, so their progesterone levels have dropped off. When do they fall? This one, day after day 25. In this one, if the oviducts are blocked, it means there's less chance of the sperm. It means that the sperm wouldn't be able to go and fertilise the eggs. A treatment. Now we're talking about IVF, in vitro fertilisation. What's happening here is the eggs are fertilised, removed, fertilised outside body, then implanted. So if the doctors were told that the person is a certain number of weeks through their pregnancy, there would be blood markers that they'd be looking for that particular point in time. If they were actually pregnant for a longer or shorter period of time, then what the doctors are checking for might make it look like there's an issue that there's not. They might then have to then go on and do other procedures to try and see if everything's okay with the baby. There are 2 tests that can be carried out with the amniocentesis or the CVS. They both have their pros and their cons. Amniocentesis has a lower chance of miscarriage. It can also be done later in the pregnancy. The CVS is done earlier in the process for that. So the miscarriage part would be what I put there. In this one, we have got their karyotype. You get genotype and karyotype. Karyotype shows all the different chromosomes present. We have sex chromosomes, X and Y, and we have the autosomes. And that's when we're talking about autosomal genetic conditions. Next one, we're talking about blood flow. We're dealing with percentage increase. We can use difference over original times 100. Let's make sure we get the correct two values. We're dealing with blood flow to heart from rest to exercise. So heart means we only care about that one. Rest to heart increase. Okay, so we're starting at 2500 and we're going to 1000. So the difference, take them away, gives you 750. Originally, at the beginning of the period we're asked, we're dealing with 250 at rest, times by 100, and that gives us a 300% increase in blood flow. For that one, we want a simple whole number ratio next. Make sure we're picking from the right column. We're asked about blood flow during exercise. So I would find the ones we're dealing with. Intestines are 500, skeletal muscles 12,000, and brain 750. We want to start to cancel this down to simplify it as much as possible. One way we can do it is, well, let's knock off 10, divide them all by 10, takes you into that. We could divide them all by 5, will fit in to that. So that would give you 10, 240. And then that would give you 15 for that. 5 again gives you 2. That would be 48. And then 3. And that would be as simple as we can get it. Another option, to be honest, probably my usually preferred option, but make this a wee bit more tricky, is divide them all by the smallest number. So we get 500 divided by itself will just be 1. 12,000 divided by 500 gives you 24. So 124. And 750 divided by 500 gives you 1.5. Now that is simple, but it's not a whole number ratio. If you double them all up, times them all by 2, that does get you to the correct answer. So at first it makes it a wee bit more complicated because it's not a whole number, but then you can double it up to get you to your answer there. So up to you, however your head works best, just follow it through and then that should work out nicely. Next one, we're asked about blood flow to these things. Okay, right, so during exercise, okay, right, so we've got 2 different parts. Now, the intestines, to do with digesting, well, we don't want as much blood flow there. So the flow to the intestines has to decrease. And these are the vasal words. So vasoconstriction means that the vessels get narrower, so less blood can pass through. Constriction. And then that means to your skeletal muscles, you have to have vasodilation, so widening of the vessels and more blood passes through. Next one, we're talking about an ECG. Calculate the heart rate during exercise. Now what we want to try and do is figure out where does one beat stop. So we go all to the top, back to the bottom, and then I'm looking for the same start part. So it looks to me, if I stop there, that looks like we are now jumping in to second beat. And then this would be the beginnings of the third beat. And if every box is 0.1, so because we've got 4 boxes, that is going to be 0.4 seconds. We want beats per minute, which is 60 seconds. I would always put a 1 in the middle. To go from 0.4 to 1, we can divide by 0.4. And to go from 1 to 60, we could times that by 60. Whatever you do to the right hand side, we need to do the same on the other side. 1 divided by 0.4 times by 60 gives you 150 beats per minute. In these questions, we're talking about sympathetic and parasympathetic. Sympathetic brings an increase, parasympathetic slows down the heart rate. We're dealing with an increase, so it's going to be sympathetic. And then in terms of the chemicals, adrenaline or noradrenaline will cause it to go up, whereas acetylcholine with the parasympathetic ones would slow it down. That's our answer there. Next one, we're talking about the capillaries and blood flow. Liquid B. So that is the part that makes up the majority of the blood, but it isn't the cells. That is the plasma. Or that the process that forms C is when we're squishing it through the walls, and that is pressure filtration. Allows the liquid to pass through it. Thicker muscle must be then an artery over a vein. Can we prove that? It's going through, yeah, getting forced through the capillary. So that would be an artery for that. So arteries, they carry blood at a higher pressure, so they need to be able to survive that. This one's to do with the how you encourage efficient exchange. So same as one before, large surface area. Or another option is they have very thin walls, which makes it easier for the chemicals to pass through. Proteins are large molecules, they will not be able to pass through the capillary system. Lymph vessel is absorbing the excess tissue fluid and returning it back into the circulatory system. Or that one, valves will be to do with the flow. They prevent the backflow. Now, we're not talking about blood vessels, so it's not only backflow of blood. It would be the lymphatic liquid, fluid, tissue fluid, backflow of fluid. Tweak it a little bit because it's definitely not blood, so make sure it goes in one direction. A sphygmomanometer or a blood pressure cuff. First thing it does is it inflates to cut off blood below, then gradually deflates. Now we're talking about systolic, so that's as the blood's flowing, pressure when blood flows again is measured. Our independent variable, the one we're changing, is to do with energy drink or not energy drink versus what we're measuring, which is the blood pressure. Okay, what have we seen happens? Look at the first values, they have not changed. And that's your systolic, so systolic blood pressure unaffected. Now the second value, diastolic value, has gone up in one group but not in the other. So in one case it's not changed, the other one it has affected it. I'd probably stick systolic because it's the same in both of them. Here we're asked to calculate their mean arterial blood pressure. Okay, so we need to do diastolic. Okay, so we're looking for group 1 after consumption. 1, 2, 3, 84. Okay, so diastolic's the second one, it's the lower of the two, which will be 84. Add on. Pulse pressure is the difference between them, so take them away from each other gives us 39 over 3. Probably, well, you could just fire that into your calculator, but it'll be 84 plus 1397 millimeters of mercury is that unit there for that. Next one, chronic sustained high blood pressure is described as being hypertension for that one. Next one, we're dealing with a study about vitamins and diabetes. Okay, equal numbers. What kind of things do we need to take into account for human trials? Well, one would be the age. Gender balance, fitness, all these kind of things. Student concluded that low concentrations increase the rate increase the risk of developing type 1 diabetes. There's no way of proving that it's the vitamin C that increases the risk of type 1 diabetes. It could be that people with type 1 diabetes have lower levels of vitamin D. So it could be the diabetes is causing the effect rather than the vitamin D causing the effect. Another problem would be it's a single study with small numbers. And this one, we're asked to calculate the number of individuals in the control group that had low concentrations of vitamin D. That is 59% of individuals. So we want 59% of 400. Percent means out of 100. So 59 out of 100 times 400. So I got caught out by that bit. If the total number of individuals is 400, that must mean there's 200 in each of the groups. So there's 200 in the individuals with diabetes and 200 in the control. So it's not 59% of 400 as I started to do. It will be 59% of 200, which is 59 over 100 times 200 will give you 118 as our answer. So I definitely get caught by that. That's how many's in the study. It doesn't say that's how many in the control group. I just took it for granted. They tried to throw me and it worked. Increasing the reliability, do more studies with more groups and then calculate the average of them. And this one we're talking about producing vitamin D in a city in Scotland. Use the data from the graph to describe the changes that occur in the average monthly hours of sunlight throughout the year. I would split this up into two parts, from January up until May, where it's increasing, and then from June through to December, where it's decreasing. Individuals need levels higher than 100. Let us take 100 and run that across. And what was the question? Which months would they need to obtain more of their vitamin C from their diet? Okay, so if it's more than 100, they get enough in their skin. But we're looking at the ones where it's less than that. I've tried to throw me, I've caught it this time. So we are looking for the ones where we're deficient in sunlight. So we need to eat it in our diet instead of just getting it through the skin. So that would be January to March, October to November. So Jan and October, December. So in the wintry months, then we'll have less sunlight. This one, we're dealing with memory. We're losing information. That's when information is replaced. We're displacing it out of our short-term memory, not being kept into the long term. Name the model that's used to explain the ability of short-term memory to perform simple cognitive function. This would be your working memory, but it can get filled up if you get overwhelmed with having to try and do too much at one time. Explain why it's difficult. You remember a mobile number in the short-term memory. So short-term memory has a limited capacity. Only store between 7 and 9 things. When I go for it, it's around 7 in piece of information they can store, and 11 is more than that as well. So it's too long to be stored by the limited capacity. Next one, we're dealing with two groups getting asked to memorize certain words. Some in categories and some randomly. Randomly allocate them. You'd have names out of a hat or use a computer program. Could be things like lollipop sticks or the spinning wheel number generators, all those lovely things that we can do for that. Why are one better at recalling them? The words are arranged into categories. They are organised, making it easier to transfer it into long-term memory. Recalling the events that occurred when information is coded into long-term memory can help the later retrieval of this information. This would be described as being contextual clues. So it might be the smell, the placement. In this question, we're looking at the combination of different neurons. We have a little gap in the neuron at P. That will be your synapse. Type of neural pathway, we've got diverging, converging, or reverberating. Diverging is when one splits off into multiple, which is what we've got here. Got the flow going that way. If it was coming together, it would be converging, and if it kept flowing back the way, that would be reverberating. How does this help with fine motor control when writing? So with the hands, a single impulse can control multiple neurons and allow each finger to move individually. Whereas if each one was done individually, then there'd be a bit of a delay between one finger moving and the next one. Coating around the nerve myelin sheath, and its job is to speed up nerve transmission. It speeds up impulses. It means information can get around the body more quickly. If you don't have the receptors, then the neurotransmitters have nowhere to bind to. And this means that the muscle won't be told to contract and carry out the movement. And in autoimmune disease, the body is attacking itself. It detects its own cells as being something dangerous. And the one that's involved in that would be T lymphocytes. When they come across their own cells, they think that they are foreign cells and they will attack them and potentially kill them. Next question. We are asked to compare the incidence of this condition. There's 20 per 100,000. And we have to work out how many more people suffer in England compared to Scotland for that. Now, to make my life easier, I'd probably look at the difference between the two populations first of all. That will be 51.6. Okay, laying it out, we've got 20 cases. If we had 100,000, but we are asked about 51.6 million. That if we go through one, so if we first of all divide by 100,000, then we times by 51.6 million. Do the same to 20,050. And that gives us 10,320 more cases in England than in Scotland. Now, we could have done that for both of them. Now, one thing that kept throwing me for a bit was the number of zeros with 51.6 million. That might be doubly problematic dealing with those ones there. So, pretty glad that I decided to take it away first and then look at what that corresponds to. But scale it down to 1, then scale it up to your number you're getting to will always work. Divide by the first one times by the second gets us our answer there. In question 13, we're asked about B lymphocytes. These are the lymphocytes that produce antibodies to help fight off pathogens. They have receptors that recognize the protein coating, the antigens on pathogens. Once they have recognized that there is an invasion, they are going to clone themselves. They produce antibodies that bind to the pathogens, which can then be destroyed by phagocytosis, by the phagocytes, engulfed and destroyed, engulfed and digested. In this one, we have a response from the B lymphocytes to harmless things. That's things like pollen, dust, and dander. That would be described as being as an allergy. If we have T lymphocytes attacking their own cells, that is described as being an autoimmune disease. Questions might play those two off against each other. Next one, we're developing a vaccine against a virus. Gene coating, antigen, gene removed, 30 purified. What does structure X represent? So we're dealing with genes, so that's going to be something to do with the DNA of that. Circular chromosome, maybe a wee bit, but it'll be DNA it's made up of. We'll go for that one. I think it's in the centre, and it's what contains the genes. For that, development of this vaccine uses parts of the virus as an antigen, say another source. If you have a pathogen that has been killed off, that can still cause a response when it's entered into the body. You can weaken the pathogen, and that makes it easier for your body to practice fighting it off. Why must they be subjected to clinical trials before being licensed for use? You don't want to put stuff that's potentially unsafe into people, so to check safety of product. Or you want to check that it's maybe better than any existing vaccines for other chemicals. So we're going to test its effectiveness. Double blind means both participants and researchers, so the people undergoing the trial or conducting the trial, do not know who's received the drug or the placebo. Now, if the virus changes the shape of its antigens, antigenic variation, then that means that the memory cells that's supposed to be able to keep an eye out for a returning virus do not recognize it. That means it's treated like a new infection where it's not able to fight it off as quickly as you would have hoped for. Last part of the paper of dealing with an essay question. We've got two choices and I'll go through how I'd approach each of them. First up with A, we're dealing with somatic and germline cells, and then talking about the different types of cell division. So somatic cells are body cells not involved in reproduction. And then in types of cell division, they can only undergo mitosis, and that produces identical daughter cells. And then germline are involved in reproduction. Mitosis of germline cells produce more germline cells. And meiosis, it's only germline that can do it, not the somatic. We can talk about diploid and haploid. So somatic germline cells are diploid with two sets of chromosomes. Gametes are haploid. As we're dealing with human, they'd have 46 chromosomes. That means the haploid, so the gametes would have 23 chromosomes. So it maintains how many chromosomes are in them. So chromosome goes back to the identical part as well. I might have covered it already. For meiosis, it involves two divisions. That's when the homologous pairs separate. Then chromatids, the individual ones, separate as well. For that, more than likely missed out some stuff as well, but that's everything I can think of to pop in there, and that'd hopefully be enough for all 8. Next one, we're talking about making the primary transcript, and then mature transcript for that. So in this one, we're going all the way back to starting off with DNA for that. So get the enzyme involved first though. So polymerase unwinds and unzips the DNA molecule. That's by breaking the hydrogen bonds between bases. Jumping back a bit, maybe put in this process. So mRNA is produced in the nucleus. Here we're talking about transcription. Then it's joining together the complementary RNA nucleotides to start making the primary transcript. Maybe worth mentioning here that RNA contains uracil and uracil in place of thymine. RNA splicing removes non-coding introns and joins together coding exons. In alternative RNA splicing, we've got different combinations of exons being kept, and makes different mature transcripts. We could show this with a diagram when we're showing that we have all the introns are being removed, and going down to the left, we're also removing exon 4. Going to the right, we're removing exons 5 and 6 as well. Importantly here that the order of the exons will need to be the same, but we can start to chop out more or less of those exons. So we could have one transcript with all 6 and with 5 of them in different combinations of the 5, and you get a whole different bunch of transcripts, which then leads to different proteins being coded for as well. If you have a question like this to try to draw a diagram, I wouldn't do as many as 6. I just stole that off a previous year's paper to fire that in. That's everything I can think of adding there. That should hopefully be enough to get all 8 marks for that question. That brings us to the end of the paper, the end of the video. Thanks for watching. Goodbye.